Okay, let's dive right into this explainer. We're going to demystify the mathematical blueprint of evolution and discover how scientists actually measure the dynamic changes of life. You look around at the sheer diversity of life on Earth, right? From microscopic bacteria to massive whales. It's super easy to just see the surface stuff, the colors, the sizes, the behaviors. But beneath all that, there's this incredible hidden world of genetics constantly shifting around. So today we're unpacking the Hardy-Weinberg principle. It's this foundational concept that actually gives us a way to mathematically prove evolution is happening, simply by defining exactly what it looks like when evolution is not happening. It's an absolutely brilliant piece of science, and we're going to break it down so it makes perfect sense. So here is our roadmap for today. We're going to start with the perfect genetic balance. Then we'll dive into the Hardy-Weinberg equation itself. After that, we'll look at the forces that disrupt equilibrium, observe natural selection in action, and finally wrap things up with the blueprint of biodiversity. All right, part 1, the perfect genetic balance, a theoretical stasis. To really wrap our heads around how evolution works, we first have to imagine a perfectly balanced scale where literally nothing is changing. This is what we call genetic equilibrium, and it gives us our baseline measurement. Picture a theoretical state where there are absolutely zero evolutionary forces acting on a population, meaning allele frequencies just stay perfectly constant, generation after generation. Honestly, it's an artificial stasis, kind of like pausing a video game, but it is completely vital for understanding the big picture. Moving on to section 2, the Hardy-Weinberg equation, the math of life. Let's translate that theoretical stasis into some actual math. Don't worry, it's a surprisingly straightforward algebraic equation that scientists use as a diagnostic tool. We start by splitting a diploid organism's genetics into a super easy binary. We're just tracking the dominant allele as P and the recessive allele as Q. That's it. That's the foundation of our equation to check the genetic pulse of a population. Just P and Q. Now, when these distinct alleles pair up during reproduction, they create 3 possible genotype combinations in the population. Let's break it down. If an offspring inherits the dominant allele from both parents, so an A and an A, that frequency is just P times P, or P squared. If they get the recessive allele from both parents, an A and an A, well, that's Q squared. And if they inherit one of each, they're heterozygous, which gives us our middle term, 2PQ. This brilliantly, and I mean perfectly, captures every single possible genetic pairing for a specific trait. So here is the absolutely crucial point. When you add P squared, 2PQ, and Q squared all together, they have to equal exactly 1. That represents 100% of the population's possible allele combinations. If a population is sitting in perfect genetic equilibrium, you can plug their alleles into this equation generation after generation, and it will always, always equal exactly 1. Which brings us to section 3, forces that disrupt equilibrium, the rule breakers. But hey, what happens in the messy, real world when our perfectly balanced scale inevitably tips? Well, if the math doesn't add up to a perfect 1, that right there is the ultimate mathematical proof that evolution is actively occurring. If observed frequencies diverge from what the Hardy-Weinberg equilibrium predicted, it's a massive red flag. But, in a good way. It's nature telling biologists that something out there in the environment is actively forcing that species to evolve. When the math breaks down, it's pretty much always because of one or more of these 5 evolutionary rule breakers: gene flow, genetic drift, mutation, genetic recombination, and natural selection. Think of these as the actual engines driving evolution forward, completely shattering that genetic piece. Let's look at the first 2. While gene flow relies on the physical movement of migrating individuals, genetic drift is all about random, mathematical chance just altering the gene pool. For example, if a group of birds flies from one island to another and joins a new flock, they're physically bringing new alleles into that group. That's gene flow. But genetic drift, that's entirely different. Drift is when allele frequencies fluctuate simply by random chance. Imagine a tree falls and accidentally squashes the only 3 bugs in a small population that happen to carry the recessive Q allele. Boom, that allele frequency just drops significantly entirely by accident. Drift is incredibly powerful, especially in really small populations where random events have a massively outsized impact. Okay, alongside migration and random chance, we also have mutations, spontaneously creating new traits, and genetic recombination continuously shuffling the genetic deck. You can think of mutations as biological typos. Sometimes they're harmful, sometimes they're totally neutral, but occasionally they introduce a brand new advantageous variation into the mix. Meanwhile,
Genetic recombination happens every single time organisms reproduce sexually. The parents' genes are mixed and matched into entirely new combinations for the offspring. Both of these processes pretty much guarantee that a population's genetic makeup is never, ever truly static.
Moving right along to Section 4, Natural Selection in Action, Reshaping the Frequencies. And this leads us to the most famous rule breaker of them all, the one that actively determines if an organism's traits help it survive or cause it to perish. See, while mutations provide the raw material, natural selection is the sculptor. It actively reshapes those allele frequencies, usually with three main outcomes. Stabilizing selection favors the average, kind of like human birth weights. Then you have directional selection, which pushes a trait to one specific extreme. Think of giraffes evolving longer necks to reach higher leaves. And finally, disruptive selection. This one actually favors individuals at both extremes while punishing the middle, which, over time, can completely split a single population to forge entirely new species. Pretty wild, right? Finally, Section 5, the blueprint of biodiversity from theory to practice. Let's look at why understanding this matters so much. It's one thing to have a neat math equation written on a chalkboard, but it's another thing entirely to use it to understand the living, breathing world. By applying the Hardy-Weinberg principle to real populations, scientists can peer beneath the surface to map out the complex processes underpinning Earth's biodiversity. Let's say biologists are monitoring an endangered leopard population. They can use this equation to track its allele frequencies over time. If they see way too much genetic drift and not enough gene flow, it means the population is getting super vulnerable. It's an absolutely crucial diagnostic tool to help us monitor genetic health and protect the natural world. Given that life is constantly being bombarded by mutations and migrations and selective pressures, it really leaves us to wonder, does this perfect mathematical stillness ever truly exist outside of a textbook? The reality is, evolution is the constant heartbeat of life on Earth. The equation almost never equals exactly one in nature over long periods of time, and honestly, that's exactly the point. It gives us the precise contrast we need to see evolution in motion. So I'll leave you with this to think about. If true equilibrium is just a theoretical baseline, are we, and literally every other species on this planet, currently